give this seminar. Thanks very much, Celia, for inviting him in the QLS series. I've known Ariel for, I think, quite a while. I think we met at Stanford, right, for the first time, a long time ago. It sounds like a couple of lives ago, but Ariel is a neuroscientist, visual perception, cognition, and a very strong one on those things. He got his education in both Stanford and Berkeley. Berkeley, I think, for your PhD, Stanford for your postdoc, if my memory is correct. And he's a neuroimager that has had a large impact on especially in diffusion imaging, but in neuroimaging in general. It's on the top of that, he's a superb data scientist and machine learner, and really has written quite a, if you listen to his talks or on educational talks on machine learning and Python and the whole Python neuroimaging ecosystem, it's really a wonder. It's wonderful. He's a very strong open science advocate, and does all his code. I don't think you have any close code in your, but you may have. But he has developed so much of the open science ecosystem tools in Python, in particular, with starting with NiTAM, if my memory is correct, and then the whole NiPy ecosystem, and of course DiPy has been a very on the a lot of your work and had so much impact on the community for diffusion imaging. And that wasn't enough. Ariel is also organizing regularly the Neuro Academy every year. It's a wonderful, absolutely fantastic summer school. If anyone has is interested in neuroimaging and data science and all those things, and it's a fantastic 2-week period that's in Seattle. That I really heavily recommend. It has a really cool way of actually accepting students with some kind of randomness, which I love. I love the system for that, because I think it's actually fairer. And engaging himself in a number of things, like startups with the INCF and other organization, of course, OHPM and so on. Ariel, it's really a very great pleasure to have you. Thanks so much, and the floor is yours.
Thanks, JB. Thank you so much for first for the invitation, also for this great introduction. I really appreciate the opportunity to tell you about the work that we're doing here. The talk today will be about what I think of as a paradigm shift in neuroscience that is still ongoing and has to do with these brain observatories that are in my title and that I will explain in just a minute. But before I go into that, let me just talk a little bit about my group. My group is the Neuroinformatics R&D group. You see the members on the right here, and we work at the intersection of human neuroscience, open source software and open data, and statistics and machine learning, sort of as JB alluded to in his introduction. And like I said, I want to talk a little bit broadly first about a paradigm shift that I see in neuroscience and actually in science more generally. I think we're moving from an era where things were, closed in some sense, where research was done as a cottage industry. Experiments were conducted in individual labs. Samples were usually small to medium-sized, and proprietary analysis tools were used to analyze these experiments. And usually in that case, in that paradigm, data was not broadly shared. I think we're moving into a new and more open model of doing neuroscience, where data collection is done in a centralized way, sometimes very large samples. I'll talk about a few samples in just a minute. And we use open source analysis tools, and data and code are often freely shared with collaborators. And this paradigm shift, I think of as a move towards what I call brain observatories. So the analogy is to the astronomy, the field of astronomy, where instruments like this beautiful picture here collect data from the sky, the night sky, and then make that data freely available. And this has been a dominant paradigm in astronomy observation for about 20 years now, and has been really fruitful in astronomy. In brain science, and particularly in human neuroscience, there have been several of these brain observatories. I think the paradigmatic example is the Human Connectome project funded by the NIH here in the US to collect data from a large sample, more than 1,000 individuals, healthy young adults, and make that data immediately freely available to the community. And that project still has a huge impact on how we do neuroscience. Other projects focused on a variety of different topics. For example, the CAMBAM sample in Cambridge focused on lifespan sampling. The Developing Human Connectome project that I will go in depth into later, that's collecting data from newborn individuals. The Child Mind Institute of the Healthy Brain Network of the Child Mind Institute that's collecting data from children and adolescents. Similarly, the ABCD, Adolescent Brain Cognitive Development Project, that's collecting also a very large sample of children and adolescents. Somebody's drawing on my slides, but that's okay. And then finally, one of the sort of models of this is the UK Biobank that's collecting really huge sample of individuals. And one of the ways to think about these very large data sets is that they compel a change in the paradigm of the science that we can do. And this comes from Jim Gray, who was a computer scientist at Microsoft and started working in database systems. And then at some point in his career, he realized that there was all this data coming from scientific fields. And he realized that this compels a paradigm change. And he called this the fourth paradigm. And that's to build on the distinct archetypes of science before, and empiricism, theory, and computation that had evolved through the 19th and 20th century. In the 21st century, we're entering the era of the fourth paradigm, an entirely new phase that involves data-intensive practices. And he called this e-science, and that's why the institute that I work at the University of Washington is also called the E-Science Institute. And this fourth paradigm unites theory, experimentation, and computation in what we now also call data science. So what can we ask with data science? And the field in which I am going to ask specific questions is in brain science and neuroscience. And specifically, I'm interested in networks. And you can go back pretty far, actually, to the 19th century to understand why we think of a brain as a network, and to the work of these neurologists who realized that specific patterns of disconnectivity, specific disconnections due to injury or due to stroke, in particular connections in the brain, causes very specific kinds of deficits, like aphasia, which is an inability to speak, agnosia, the inability to recognize, alexia is a specific deficit in reading. And they inferred from that that specific brain connections have specific functional significance. And this is an idea that has evolved now to this notion of a connectome, the idea that brain connectivity, or the brain as a network, is what defines the function of the brain. And so we're interested in studying brain networks. One of the important components of the brain networks are, of course, neurons. So this is a neuron, and the cell bodies of neurons, they lay in the gray matter. So this is a human brain postmortem dissected out so that we can see the cell bodies in this thin layer in the cortex, and then the axons that connect, that communicate between one neuron and the other. The axons of the long-range connections between brain areas are here in what's called the white matter. The white matter is this region in the brain that contains all the wiring, the connections between different parts of the brain. And the reason that it's white is that these axons are covered in a sheath of myelin. Myelin is a substance that insulates the axonal connections, the electronic transmission in the axons, and makes it more energy efficient, more accurate, and more rapid. And that really is important. The health of this part of the brain, the white matter, is really important for brain function more generally and brain health. And one of the ways to think about this is to just look at the dynamics of myelin development over the lifespan. And so we see waves of development and waves of degeneration in early childhood, in childhood and adolescence. And then in adulthood, myelination of the brain stays constant for a long time. And then in aging and senescence, there's the deterioration again of the myelin. And these waves are important because they also affect brain health. So if the dynamics of myelination, something happens to the dynamics of myelination at any one of these stages, it can affect different kinds of disorders. For example, if early childhood myelination doesn't function, then that can lead to development of neurodevelopmental disorders. Psychiatric disorders usually arise in adolescence. They can also be related to deficits in myelination during this period. And then, of course, neurodegenerative disorders and aging are also affecting the myelin, the ability of the brain to communicate. How do we measure this? You can measure the properties of the myelin in people, in living humans, using magnetic resonance imaging. This is pictures of the MRI machine that's actually in the building, in the basement of the building that I'm talking to you from right now. MRI is really an amazing field of technology. One of the amazing things about MRI is that it gives us millimeter resolution images of the human brain in vivo that are sensitive to different physical brain properties. So here, for example, on the left is a T1 weighted MRI, on the right is a T2 weighted MRI, and those are different ways of measuring brain properties. The MRI aficionados will notice that these are quite strange T1 weighted MRIs and T2 weighted MRIs. And I'll come back to these images later in the talk to explain why they look so strange. The method that we use is called diffusion MRI. In diffusion MRI, we make the MR image sensitive to the direction of diffusion of water within the tissue by aligning diffusion gradients or applying pulses of diffusion, or sorry, magnetic field gradients along the sample. And what you need to take from this is just that we can get different images that are sensitive to diffusion in different directions within every voxel, every sampling voxel within the brain. And if we make the measurement sensitive to diffusion in different directions, in places where there's isotropic diffusion, where diffusion of water can be freely available in all directions, then we get a similar signal in all directions. But in places where there are axons, where there are these myelinated axons, here's again another picture of an axon, this time an electron micrograph and a cross-section through the axon. What you can see here is the myelin wrapping around the axon. So in the middle here is the lumen of the axon, and around are these myelin sheaths. And what these myelin sheaths do is that they form these bundles. So axons travel together in bundles. Here's another microscopic image of a bundle. And here's actually macroscopic image of a big bundle. These are the optic radiations that transmit visual information from the thalamus back to the end of the cortex. And within these bundles, what happens is that the diffusion of water is anisotropic. Water can more readily diffuse along the length of the bundles than across these myelin boundaries that I showed you wrapping around the axon. And because of this property, we can derive all kinds of properties of the tissue from the measurement of diffusion MRI. For example, we can derive the mean diffusivity. How much diffusion is there? How restricted is the diffusion? And diffusion is very restricted in places where there's a lot of tissue impeding the motion of the random motion of molecules of water. In some places, diffusion is more readily available in some directions because of the orientation of these bundles. And we quantify this through something that we call fractional anisotropy. This is a number between 0 and 1. If it's 1, that means that diffusion is really restricted to some directions and it's not available in other directions. And we can also infer things about, for example, the principal diffusion direction, which tells us in what directions diffusion can freely happen. For example, if we look at the corpus callosum, which is a big bundle that connects the two hemispheres, diffusion is mostly available in the right-left direction. But if we look over here in this blue area here that encodes the up-down direction, this is in the corticospinal tract. And so there's a big bundle that's traveling up-down in this region, and that's why the color is blue. It's encoded as red for right-left, blue for up-down, and green for anterior-posterior, like these fibers or these bundles in the front of the brain. Now we can use these properties in order to do what we call computational track tracing. Track the connections between different regions of the brain in a living individual. For example, if we look at a horizontal section through the brain of one individual here, and we zoom in on this particular location, we can model the diffusion, the directional diffusion, every location within the white matter, and then place a seed in a particular location. For example, if I place a seed in this particular location, and I just follow the principal diffusion direction, propagating a streamline through this kind of vector field of principal diffusion directions, I might get a streamline, a line that connects between the two hemispheres, again, through this corpus callosum, this bundle that connects the two hemispheres. But if I put a seed and I start propagating a streamline from a place close by, I might get a streamline that goes to the back of the brain. And if I repeat this process hundreds of thousands of times, I'll get many, many streamlines here encoded again as their color indicating the direction of the streamlines throughout the entire brain. Now, before we're interested in the functional units of the brain, the functional units that these neurologists identified. And so we'd like to divide the connections into different functional connections. And here is the kind of picture that we'd like to get, where we get different parts of the brain or different connections that have functional significance. For example, I mentioned the corticospinal tract. Here it is in orange in a single individual from the Human Connectome Project, in this case, traveling up down from the motor cortex down to the spine to control volitional movement in muscles in our body. So in order to get that from this set of streamlines that we tracked, we do what we call computational dissection. We say, look, there's a region here in yellow on the right, and I would like you to give me all the streamlines from this large collection of streamlines that pass through this yellow region. And when we do this multiple times for multiple different regions of interest and connect this together, we call that automated fiber quantification. Through automated fiber quantification, we can identify these functional units that I mentioned. This is a multi-step process where we do whole brain tractography. We do segmentation of the streamlines that correspond to a particular bundle. We do some refinement of this and then cleaning. And then finally, we quantify the tissue properties along the length of the fiber by sampling the tissue along the length of the fiber. So here's an example. This is the arcuate fasciculus in the left hemisphere of one subject. And you can see that there is, in the color map here in yellow, are regions of high fractional anisotropy. So we get these kinds of track profiles. These are one-dimensional kinds of vectors that tell us along the length of this bundle, what are the tissue properties, in this case, fractional anisotropy. And this is what we quantify and try to assess. And we've developed open source software called PyFQ. John Kruper, who's a student in my group, in collaboration with Jason Yateman and his group at Stanford. And we've been developing this for several years and make this software freely available to anyone who wants to use it and do research of this sort. So what can we measure with diffusion? One thing we could do is measure the biological basis of neurological disease. For example, amyotrophic lateral sclerosis, ALS, also known as Lou Gehrig's disease. This is a disease that affects the corticospinal tract that I already mentioned. And one way that we can assess this is by comparing a group of patients with ALS here in blue and their fractional anisotropy along the length of this tract with a group of controls here in red, and find very significant and consistent differences between these two groups. Another thing that we can measure is brain development in childhood and adolescence. For example, so in this period of time that I mentioned as a wave of development, for example, in this very large sample that I mentioned before, the Healthy Brain Network, which is a project of the Child Mind Institute. And so in this case, we're looking at multiple bundles throughout the brain, right? In the ALS case, we looked at just one bundle. Here, we're looking at multiple bundles. And so here are multiple track profiles. And here are the developments, each bin here. So we start with blue as young children and yellow as older children. And what you can see is that throughout the entire brain, FA is changing through this time, and that's because of the increase in myelination that's indicated here in this schematic on the right. And at the same time, mean diffusivity is going down, so from blue on the top to the bottom. And we see this throughout the entire brain. So in some cases, we study specific connections. In other cases, we can study the entire brain. And so now I'm going to focus on a particular study where we ask what happens to brain connections and their tissue properties at birth, right at birth. And to study this, we used this data from this particular brain observatory called the Developing Human Connectome Project. They've developed methods to measure diffusion MRI and other kinds of MRI in infants in the few first days after birth, which is quite incredible. And so one thing to think about is here as an image from my collaborator, Marika Groth. Here is a picture you'll see in just a second. For now, I'm just showing you her brain on the right, and then an infant brain on the left. Infant brains are very, very different from grown-up brains in many, many different respects. And so we needed to develop specialized software to deal with that. And that's developments done together with Marika and her team at the University of Marburg. And also I should mention Jason and Kalnit at Stanford and members of my group who worked on this project. developing this. And we just recently published this, the paper that describes the first of the projects that depend on this software. So here you're seeing an infant brain on the left with a few of these bundles that I described before, delineated in the baby brain. So we'd like to ask in this project, what happens right at birth? And there's a few different theories. We know that the myelination starts in utero from large body of previous work and continues to happen after the baby is born. But the question is, what happens right at birth? And one theory you might have, one hypothesis you might come up with, is that there's constant development. Myelination just continues at the same pace. Another model you can have is that as soon as the baby is born, they're starting to be exposed to more stimuli, their eyes open, they're starting to interact with their social environment, and there's accelerated brain plasticity and accelerated ex utero myelination. And then finally, the third option or third hypothesis you might have is that as they're born, they go out from this sheltered environment of the womb that they were in, and there's a slowing down of the myelin development. In order to study this question, we looked at this sample of babies from the Developing Human Connectome project. Let me just first set up some terminology that I will use. The age of the baby, actually, whenever their MRI scan is taken, that can be defined using several different periods. One age that we look at, this is the post-menstrual age. That's the time since the last menses. So that's the age of development in utero, and then also the time that passed from birth to the scan. But that can also be divided in two separate periods. The gestational age is their post-menstrual age at the time that they were born. And then their scan age is their age at scan taken from birth. And we define this using, weeks. So we looked at a sample that's a cross-sectional sample first. These are measurements from 300 infants born between 25 and 42 weeks of gestational age, and scanned 29 to 44 weeks post-menstrual age, which was 0 to 16 weeks scan age after they were born. So you'll see there's a large range of gestational ages. Full term is 40 weeks. So 25 weeks is very, very early preterm birth. The marker that we looked at for myelin is the T1 weighted over T2 weighted ratio. So this is the T1 weighted image that I showed you before. And the reason that it's strange is that it is taken from an infant about 1 week after birth, and the brain just looks really, really different. Those of you who've seen grown-up T1 weighted will notice, for example, that the cortex here is brighter than the white matter, which is the opposite of what we see in grown-ups. Here's the T2 weighted image. And these two images are sensitive to myelin, but it turns out that they're sensitive in opposite directions. And so if we divide one image by the other, a trick that Matt Glasser and others came up with, we get this image that's sensitive to myelin, and we use this as an in vivo marker of myelin. And we can look at this ratio image, this marker of myelin, along the length of different bundles. So here's just two bundles, the frontal corpus callosum and the posterior corpus callosum. So these are three different babies at three different ages. And we're dividing our group of 300 babies into three different bins here. And we're looking at just the evolution of myelination in those first few weeks of life. And what we can see is really massive myelination in early life. So myelination is very, very rapid in early life. That's the first thing to note here. And it's rapid throughout the brain. So here I'm showing you many different bundles. Again, on the left here are images of bundles in a younger baby. In the middle is a slightly older baby. In the right, an even slightly older baby. But you'll notice these are a span of just a few weeks, from 33 to 45 weeks roughly. This is post-gestational age. And you can see that there is really rapid myelination throughout every one of the bundles that we've looked at. So again, we'd like to address this question, what happens at birth? And one way that we can address this in this sample is to develop two kinds of models. In one model, we would say that the only thing that affects how much myelination there is in an individual part of the brain, an individual node along a particular track, is the post-menstrual age. just the span of time that has happened since the baby was first conceived. But we can have an alternative model that says that there are two factors here. One factor in the model is the gestational age, and another factor is the scan age. And this model says that there are actually two different rates that we need to think about. One rate in utero, the period of gestational age, and another rate that we need to think about, which is the ex utero, the time that passed since the scan. And this model, or comparing these two models, will allow us then to adjudicate between the constant rate and the changing rate models that we had on the previous slide. And what we found when we're comparing those two models in the data is that overwhelmingly the two-factor model, the two-component model, fits the data better in all parts of the brain, pretty much. So we can, at the very least, we can exclude this first model of constant development, the one that is represented by this equation on top here. So the question is there faster ex utero development or slower ex utero development? And this we can answer by looking at the different coefficients of the model. So here we have, again, all the different bundles, and along the length of each of these bundles, we've now plotted the slopes, these beta coefficients of the linear model, where the solid line is the coefficient for the scan age, and the dashed line or the dotted line is for the in utero development, the coefficient for the gestational age. And what we find is that overwhelmingly in all parts of the brain, pretty much the in utero development is more substantial than ex utero development, suggesting that the correct model is that there is a slowing down of development as soon as the baby is born. So next we might ask a question about preterm birth. So preterm birth has a significant impact on cognitive abilities, on cognitive development in general. And the question that we'd like to ask is whether this slower ex utero development we see early in life accounts for effects of preterm birth, at least very early. So first of all, okay, so how do we do that? In addition to the 300 babies that we looked at in the cross-sectional sample, we also have a longitudinal sample from the Developing Human Connectome project. These are measurements from 34 infants that were born preterm. On average, so preterm is defined as 37 weeks or earlier, and these babies were born on average at 31 plus or minus 3 weeks gestational age. And these babies were scanned twice. They were scanned once at when they were born, roughly when they were born, so roughly at 34 weeks post-menstrual or post-gestational age, and again at what we call full-term equivalent, at approximately 41 weeks post-menstrual. And this allows us to compare babies that were born preterm, scanned at full-term equivalent, with measurements from 34 matched infants that are born full-term. So we matched from the cross-sectional measurements that I described here. We initially took and matched 34 infants in terms of post-gestational age and sex, so that we had a matched sample. We had babies that could be compared on an equivalent post-gestational age. And we actually set those aside and didn't use them in the cross-sectional sample at all. And that will become important in what will follow. So we have 34 full-term born infants that are matched to these 34 infants that are born preterm, and we can compare them directly. So what we have here is 3 lines. The solid line is the preterms measured at birth. The dashed line are the preterms measured later at scan age of larger than 37 weeks, so at full-term equivalent, and then and dotted line, the full term. And what we notice here is 2 things. First of all, in babies that are born preterm, we see similar, very rapid development early in life. So the solid line is much, much lower than the dashed line. This is just within those longitudinal preterm born babies. But the other thing that we should notice is also that the babies do not catch up with the full term, the babies born full term at this full term equivalent. That we notice because this dotted line is higher than the dashed line. So the babies born preterm are delayed in terms of the their brain myelination, and they're delayed early in life. So the question is, because of this gap between ex utero and in utero development that we established in the cross-sectional sample. In order to test that question, we can take the first measurement from these babies where longitudinal measurements were done. We can put it through the model that was fit on the cross-sectional sample, and we can then predict the measurement, the second measurement, the measurement that was done at full-term equivalent. And, oh, sorry. And then we can compare that to the actual measurement that was done. So we can predict that measurement using this model that was fit on a completely different sample, and then compare that to the second measurement. And so what we have here is just the data from the longitudinal babies that were born preterm, and the measured data is in color. The constant developmental model here is in dashed lines, and the in utero / ex utero model, the model that posits two different rates of development for in utero and ex utero, is here in dotted. What you can see when you look at the graphs is that the dotted lines follow the colored lines a lot more closely. And we've also quantified this. So the model that posits constant development doesn't fit the data quite as well as the model that posits that there are two different rates of development. So in fact, the slower ex utero development model explains the data and explains this delay in development in babies that were born preterm. Okay, are there questions? I see somebody drawing here, so I'm just gonna stop here and see if there are any questions about this data. Okay. So our conclusions from this are that there's rapid myelination very early in life, and there's slower myelination ex utero than in utero. And this slower ex utero myelination explains the myelination gap in infants that are born preterm. What does that mean? Well, at the time of birth, there's a change in sensory input. We know that the baby is first exposed to all kinds of sensory input that is very, very much dampened when they're in utero. And so there might be some experience-dependent plasticity, but at the same time, there's a change in nutrition. And infants, in fact, lose 5 to 10% of body weight in the first 2 weeks of life. And so we speculate that the change in nutrition, due to the transition from the nutrition that comes from the umbilical cord to feeding on their own, that change in nutrition outweighs the impact of experience-dependent plasticity in these first few weeks of life. And this has potential implications for interventions and assessments in cases of preterm birth. One really extreme case for that is from recent news. So this is not our work, but the work of others in this really remarkable paper from Partridge et al. In 2017, where they designed an artificial womb. And this was recently in the news, and that's because the FDA, the US FDA, is considering how they might do clinical trials of this kind of system that mimics the womb. Obviously here, not in a human subject, but they're considering how they might do something like this in humans as well. And what you can see, this is a system that stimulates the womb, including the nutrition coming in here through a tube straight through the umbilical cord. And so this might become a reality. Our research kind of underscores why you might want to go ahead with these kinds of rather complicated trials they're about to start. Okay, so actually I'll just wait here for a second, since I see that I'm ahead of my planned time, and there's still a few things that I want to tell you about, but I'm just going to stop here and see if there are any questions about this particular research project that I described. If no questions, then and you can,
yeah, maybe
you can
I can ask one quick question for you.
Yeah.
How many infants were involved, and do you know if other factors, because there's genetic factors, there's environmental factors during the pregnancy, there's maybe BMI of the mother, whatever, there's so many other factors that could influence those development aspects. Is there, have they looked at that, and what's
Yeah, this is a really great question. I think it also I think it points to a challenge with these brain observatories. At the time that we did this this study, we only had about these babies. The only things we knew about these babies were their various the ages that I described, and so the age at scan and the age at kind of post gestational age at birth, and their body weight at birth, and a neurological score, some kind of like an APGAR kind of test that was done at birth, and their sex, and whether they were twins or not. Those were the only things we knew because that was the only data that was released together with that original release. And you might ask, well, does the neurological score, the body weight tell us more? And the answer is that correlates really, really highly with the post-gestational age at birth. Babies that are born preterm are also very low body weight. And so we didn't have a lot of this kind of information. But now the Developing Human Connectome Project has in fact released a lot more information, including a lot of the questions that you asked. And so we're going to continue to study. In fact, they've increased the sample. Now there's, I think, 800 infants in their third wave or third release that we are currently studying and continuing to study and ask questions like the ones that you asked. Okay, so let me zoom out and we can come back to talk about this study and other things later on. Let me just zoom out to this paradigm shift that I described in the beginning and just ask a little bit, what do we need in order for this paradigm to work well? Once there's data of the sort that these brain observatories are releasing, what other components need to be in place in order for this to work well, for us to be able to make important discoveries, to make progress? I'll claim that there's what I'll call an ecosystem of open science that needs to exist. And the first component of that for me, and maybe I'm biased because that's what I do, is open source software. So I described this open source software that we developed called PyFQ, and we have a sort of a complement of tools that we developed that others can take and use on other samples, and they can explore the analysis that we did. For example, in this baby case, we released all of our derived measures, the track profiles, and so on, so that others can study these and ask additional questions from the data. But our software doesn't live in its own sort of bubble. In fact, it relies on an ecosystem. For example, we use methods that we and others have developed into this broader ecosystem of DIPy, diffusion imaging in Python. And here I'm mentioning Eleftherios Garifalidis, who's my collaborator, longtime collaborator, more than 10 years that we've collaborated on developing the software, and Serge Koudoro, who's a software engineer at Indiana University, who leads the development of DIPy. But DIPy itself also depends on an ecosystem, the neuroimaging in Python community, a broader community of people who do software development in Python for a variety of neuroimaging applications. And we learn from those people, and we depend on a variety of other components that they develop for everything that we do. But in turn, that development depends on broader developments like the NumPy community, the SciPy community, of course, the open source software community around the Python language in general. And I like to quote Brett Cannon, who's a Python core developer, who famously said, "I came for the language, but I stayed for the community," which really tells you a lot about how we learn from others within this community and rely on the work of others and kind of this network of interactions with others in the community. But an ecosystem of open science doesn't only comprise of the software. In order for us to be able to do work, we need to be able to speak a common language. And that common language is the language of standards. Open source standards and the development of standards that support a community of researchers is exemplified by this project called the Brain Imaging Data Structure. You may have heard, those of you who do neuroimaging may have heard of BIDS. BIDS is a fantastic project that's been around for about 10 years now and sets standards for how to arrange and describe data sets of neuroimaging. And since its development, it's also expanded into other modes of data collection. It's constantly expanding. And I'm actually excited because, well, I'll just mention this is a huge community with over 300 contributors worldwide from many different institutions. And the specification, the website that holds the specification has had 10,000 unique visitors just in the first half of 2023. And now the thing that I'm excited about is that the BIDS community was just awarded the Neuro Irvin Helga Cooper Foundation Open Science Prize for 2023. And so those of you, all of you actually who are in Montreal can attend the Open Science Symposium on November 30th and hear more about BIDS from Yaroslav Halchenko, who will represent the project there at this event. Okay, and then JB in his introduction mentioned my passion for education or for training. And I think that really an important component in making all these things work, standards, software, data are really good. But if the people who are supposed to use, students and researchers who are supposed to use these tools, these data, don't have access because of the knowledge that they require, then it's hard to make progress. My own contribution here is really inspired by a thing called brainhack.org. Brainhack.org is an organization that organizes hackathons. Hackathons are kind of short, intense periods of time where you work together with others and collaborate. They exemplify this spirit of community that I mentioned before around open source tools. And based on the idea of brainhack.org, Brainhack organizes hackathons, for example, the annual meeting of the Organization for Human Brain Mapping. And based on kind of inspired by this model, Talia Arconi and I started an event that we call now Neuro Academy. Neuro Academy is a 2-week event. In the first week, we teach a lot of things with many instructors, including JB and others, who teach a variety of ways in which data science integrates with neuroimaging data, various methods. We teach programming in Python, and we teach a variety of machine learning methods and a variety of different kinds of software libraries and things that you can use in order to analyze these kind of brain observatory data. You mentioned that Noah Benson, my colleague here at UW, Talia Arconi left the world of science and joined the world of industry a couple of years ago. And Noah Benson, who's my colleague here at UW, is now my collaborator on Neuro Academy. And we will have Neuro Academies this year and until at least 2027, thanks to a grant that we have from the National Institutes of Health to do this. So if you're a student or a postdoc or anyone really can sign up, go to this neuroacademy.org website. We'll probably open applications around early in 2024. And in fact, in the spirit of disseminating all these ideas, Tal and I wrote a book called Data Science for Neuroimaging: An Introduction. This book will come out in December. I think the ebook is already available, and you can also find the content of this book on this website, neuroimagingdatascience.org, that includes a lot of code and data and various explanations about the kinds of approaches, the data science approaches that we take in analyzing brain observatory and other imaging data. And I just wanted to also highlight that this kind of idea of open source education, and one of the benefits of these brain observatories, these open data, is that researchers who work in environments where data collection is really difficult because of a variety of constraints, including cost constraints, MRI machines are just really expensive, can actually download these data and work with them. And that opens up a lot of possibilities. I mentioned Damien Eek because he started the African, or he's one of the people who started the African Brain Data Network. And I'm really excited that they're having a summer school a little bit similar to Neuro Academy in just a few weeks that I will give a lecture at. So I thought I'd mention that because it's a cool project and I'm excited about it. Okay, and now I'm really ready to close. I just want to thank all the sources of support for the work that I described from various funding agencies, and then mention Marika again, who led the work on the infants that I showed you. And that's the website at the bottom is the website for my group, and you can go there and learn more about our work and other projects that we've worked on, papers, and who's in the group and so on. And I'm happy to take questions now.
I'm not sure if I am the one monitoring the questions, Celia. Don't know what's.
Yes, please, JB.
Okay. Actually, want to see if everyone maybe infused. Yeah, I mean, I guess anyone, please chime in and ask Ariel's question if you have. I can't see everyone on the screen, so I might miss some hands raising.
Feel free to put a question into the chat also, and we'll
there we
keep an
go.
eye on it.
There is
Okay, so
actually
there is a question in the chat, and I'll just read it out and then answer it. The question from Benjamin is, thank you very much for your presentation. Thank you. I have a couple of questions about tractography. For your comparisons, did you manually select homologous fibers, or did you use some sort of statistical atlas-based approach? Do you think that AFQ can be applied to vector fields produced using processes other than diffusion MRI to identify fibers and or bundles, not necessarily in brain imaging? Yes, so I'll go back to this slide here, because I think that if you're working with other kinds of data, PyFQ is sort of a solution for a very particular kind. It's a pipeline. It starts with diffusion MRI data organized according to this standard, the BIDS specification, go through a series of steps. To answer your question, it uses something like an atlas-based approach. The atlas-based approach just says, if I want to find the corticospinal tract, find for me all the streamlines that pass through some area down at the bottom here and another area up here. Compare that then to a statistical map, like a probabilistic map derived from a large population, and then that will be my bundle. So it does use this statistical atlas-based approach, which is very specific to the brain. But all of the tracking methods that are underlying that and that are implemented in DIPy, those can actually be applied, as you say, to other kinds of vector fields. In fact, not only vector fields, but also fields where there are multiple different directions in every voxel or pixel. And we use those kinds of methods in places where there's crossings of different bundles. And so that you can take and you can apply that to other kinds of data. And people have applied this to a variety of other kinds of data. We've seen people use this in microscopic data, and even recently to geographic data where there's sort of roads and things of that sort.
Thanks, Ariel. Any other question for Ariel? Actually, might chime in with my own question. Like, I think those observatories and big data sets are obviously wonderful, but at some stage there are also many labs acquiring other data sets that are related. And what do you think are the hurdles, or what are the problems that are neuroscientists facing when they're trying to co-analyze all those things? And what do you think is the right direction to go in that area?
Yeah, I think maybe my presentation was a little bit too stark, where I said there's this old way of doing things and this new way of things, and we need to, maybe my implication was that we need to get rid of the old and only do the new, and I think that's not the case. And that's also Jim Gray, who presented this fourth paradigm. His idea was that this would be an additional paradigm on top of empirical and theoretical and computational studies that have been done in the past. And I think there needs to be a cycle of generating hypotheses using these brain observatories and then testing those hypotheses into new experimental data. I think experimentalists of the kind who do this kind of cottage industry, like specific experiments, they are in a much better position to ask much more specific questions that arise from the data. For example, just kind of a very contrived example maybe, but one that comes from our own work, is that if your hypothesis is that nutrition early in life is really important for myelination, some experimentalist might do an experiment where you do some kind of intervention related to nutrition early in life. That's not something that we can do with the brain observatories, but that might be something that someone in an experimentalist doing studies on infants can do. Or ask more specific questions. You had more specific questions about, for example, the various factors or kind of in utero factors that might affect myelination. You might collect a particular sample. For example, my colleagues here are interested in effects of cannabis use. Cannabis is legal in Washington, so there you might collect a specific sample of mothers who have consumed cannabis and a control sample that matches that. That's the kind of question that's very hard to ask with brain observatories, because you might not even have that kind of sample.
I have a question myself. So when you're estimating, tracks or whatever, are some of them sort of much easier to capture than others? And sort of that inherent difference in precision, how do you take that on and carry that forward in next stages of analysis?
Yeah, that's a really good question. And it is true that some of these bundles are harder to find than others. John, who I mentioned earlier, actually, we just had a paper in a workshop, a MICAI workshop, talking about the optic tract, which is the bundle of fibers from the eye to the thalamus. And that's really small. It's part of the brain that has multiple other kinds of things like muscles. The eye muscles are right there. And it's a lot harder to track. And so we needed to develop specific methods to find that particular place. I'll admit that in our statistical analysis, once we're done with delineating this thing in a robust way, in a way that we consider robust, we don't consider that a challenge to precision. But now when you're asking it, I think that maybe we could. A lot of the bundles that I showed you before are equally hard to find. So for a lot of the stuff that we've done so far, that's not a huge challenge. As we go to smaller bundles, we also ask more specific questions. The reason we're interested in the optic tract is we're interested in retinal disorders and how those affect. There, we might ask a question about that particular tract, not comparing necessarily across tract, but looking at that particular tract, say in individuals with retinal disorders and individuals who don't have retinal disorders.
Then maybe it doesn't matter so much unless there's a lot of individual variability in your ability to capture that bundle.
Yes.
Right. So maybe
we definitely worry if somebody has.
Yeah, like if it's actually associated with disease or phenotype, then that's where the issues might come in. Or
Agreed. Like if somebody has a retinal disorder that affects those fibers, and for example, in glaucoma or age-related macular degeneration, there might actually be loss of those fibers. And then they would have a smaller bundle and maybe different tissue properties as well. And the tissue properties are not entirely independent of our ability to track. Where there's higher FA, we also have a clearer signal for the tracking. And those are always concerns. We always try to figure out ways to equate between the groups in terms of the difficulty of finding the bundle. In some sense that it's okay if we can find where that bundle is in the individual. Now, for example, when we look at the optic radiations, which is the bundle from the thalamus to the cortex, every individual who can see has an optic radiation. There's no question that they have an optic radiation. The question is just where is that optic radiation and can we characterize it? Every person who can control the muscles in their body has a corticospinal tract. And so individuals with ALS, for example, might have some damage to the corticospinal tract. But in their development, they develop the corticospinal tract. We can find it in every individual and we can compare the two groups.
So it's less of interest to quantify the size or strength or what I don't know quite what the right adjective is of that bundle than it is just to talk about presence absence.
Yes, I agree. And we focus on these tissue properties because we are a little bit wary and skeptical of, for example, looking at the volume of the bundle or the number of streamlines within the bundle, because there are other reasons, kind of like confounds, that might affect the volume, might affect the number of streamlines. So our focus is on the relatively robust, and we've quantified this robustness in work that John did a couple of years ago, relatively robust measurements of the tissue properties along the length of the bundle.
Thank you.
Any other question? Maybe a last question, if you
from the seminar room want to step up to turn on the microphone and ask a question?
I can't see.
Yeah, I think right now it's
Okay.
We can't.
Maybe last question then, Ariel. You developed a lot of open source neuroimaging software, and what do you think are still the key challenges, and what would you like to see happening? In which direction should we work to make those sustainable and evolving in the right way? And without, I mean, a bit of general questions, and hopefully.
Yeah, with a thorny topic. So I'll say just off the top of my head, one of the things that we need to develop are metrics that support people who are interested in developing open source software. One of the challenges are the career paths of people who develop open source software, right? I work with students, I work with postdocs, work on these things, and they ask themselves the question, okay, what do I do after that? If people will only look at my record of publication of papers in high impact journals, what can I do that to sort of support my career if I'm interested in developing? And so I think we need to think of alternative career paths about career. We need to think of career paths within universities and research institutions that award specifically, kind of reward specifically, and advance people specifically for their contributions to these kinds of projects. I think that's something that's currently done piecemeal. University of Washington in particular has been really great. My own career here started as someone in that, and kind of I was able to advance in my career because the University of Washington thinks about these things. But we need, I think, broader policies at the level of funding bodies, at the level of journals, at the level of broader institutions. On a similar note, asks Benjamin in the chat, can you recommend any resources on how to write and distribute good open source scientific code that people can use, modify, and maintain? So I will point to my own work. In the Neuroimaging Data Science book that we wrote and that you can find the content of in that website, we do have a chapter about how to develop, kind of both on the technical side, but also a little bit more about the broader collaborative aspect of this. I'll also mention the Journal of Open Source Software, which is a journal devoted to open source software that I think does really well in describing the standards and the best practices. So when you publish there, there's a list of things that the software needs to include. And I think that does really well in sort of promoting best practices in scientific software.
Cool. Any other question for Ariel? I can't see any. Ariel, it was wonderful. Thank you so much. It was really a pleasure, and it was super didactic and super pedagogical as well in a number of areas. So really, thanks so much for that. And yeah, hope to see you soon. Or
yeah,
you.
thank you all.
Thank you so much.
And hope to see you all soon. Thanks so much. Bye.
Bye-bye.
Yeah.